This video is about protein synthesis, that is, the process by which proteins are made. To understand that, we have to first look at RNA. There's three different kinds of RNA: messenger, ribosomal, transfer. Messenger RNA is going to copy the DNA message, carry that message out of the nucleus and to the ribosomes. Ribosomes are just made of RNA, ribosomal RNA. And what they're responsible for is assembling proteins and getting that amino acid chain linked together. Transfer RNA is responsible for transferring the amino acid that they carry to the growing protein chain. So let's take a look at how some of those things work. First, here are the three different types of RNA. You'll notice that messenger RNA is a long strand, ribosomal RNA is in the shape of a ribosome, and transfer RNA kind of has this odd looking T shape to it. When you look at messenger RNA, it's read three nitrogen bases at a time. That's called the codon. So think of it like the code. So code, codon, three nitrogen bases at a time. So for example, if that codon is A, the messenger RNA attaches to the ribosomal RNA, and the ribosomal RNA is going to start to collect transfer RNAs until it finds one that fits the A codon. So that codon has to be matched up or paired up with what's called an anticodon, another set of three that's on the bottom of the transfer RNA. If those three pairs don't match, then that amino acid is not added to the chain. So what matches up with AAA? Well, normally what would match up is a TTT, but that's just for DNA. RNA doesn't have TTT. So in this case, we're looking for a UUU. So if the transfer RNA has three uracils, it will transfer that particular amino acid to the chain. And that's how the amino acid chain is formed based on the instructions of the messenger RNA. Here's kind of what that looks like. In the nucleus, we have the DNA strand being copied by the messenger RNA strand. That's transcription. So think of transcription like a scribe is copying some foreign language. Translation is when someone takes that copy, looks at the foreign language, and turns it into something that you understand. In this case, an English sentence. So here we have transcribing, which is just the messenger RNA being copied. The messenger RNA leaves the nucleus to the ribosome. So this is happening in the cytoplasm now. The codons are being read by the transfer RNA so that the amino acids can be added. Well, how could we know, based on the codes, how do we know what amino acid is next? Well, we can figure that out by using charts that look like this. All right, so if the codon is CUG, we can figure out what amino acid that is. So first, we start in the center at the C. Then the second letter is U, so we just move out to the middle ring and find the U. Then we move out to the final ring for the G. So CUG would be leucine. You'll notice that some amino acids are special, like AUG is for a start codon. That's kind of how, it's like a capital letter in the English language. It's how the protein begins. These, with the dots here, represent stop codons. That's how we know the instructions are complete. The message is done. So whenever there is an AUG code, that terminates the message. Another chart looks like this one. You just read it in a very similar way. This is the first letter. So for example, if the codon was CUU, then I would start here at the C. I go over to the second position from across the top, find a U, and then look for that third letter U. So CUU, that's leucine. You'll notice that there are a lot of combinations that give us the same amino acids, because there's only 20 amino acids. The good news is oftentimes if there's a mutation, the result is silent. There is no dramatic thing that happens because CUU, if that's mutated to CUC, there's still leucine in the amino acid chain, and the message comes out loud and clear. So I hope that helps you understand a little bit more about transcription and translation.